The CFDE C2M2 Fair Data Package Assessment Appyter, built in collaboration between the LINX DCC and the CFDE-CC, performs fair assessments on C2M2 data packages from each DCC using metrics that align with the C2M2 metadata model specification. The CFDE aims to adhere to the FAIR principles, which stand for findability, accessibility, interoperability, and reusability. To enable participating Common Fund DCCs with the ability to evaluate the fairness of their data packages, we developed the C2M2 Fair Assessment Appyter. This appyter specifically measures the conformance of an input data package with the C2M2 metadata guidelines. The assessment focuses on the different aspects of the C2M2 model which make DCC data packages fair. Users can upload a zip file containing a valid C2M2 data package, and the appyter will do the fair assessment automatically. First, the data package is loaded, and global metrics are computed. In this table, all entity counts are shown for the submitted data package. Here, we can see that the data package includes data from 19 different anatomy labels, 40 assay types, about 1,300 biosamples, and so on. Then, the appyter computes compliance with the C2M2 specifications. The overall metadata coverage is broken down by the table, where each value gives information on how many of the C2M2 specified columns are filled in for each entry in a table. For instance, the first row tells us that on average, about half of the C2M2 specified metadata fields are filled in for the project table. The appyter proceeds to evaluate the data package against the C2M2 model guidelines. The appyter will check whether file persistent identifiers follow a valid schema. In order to maximize findability, file identifiers should be resolvable and unique to the specific file. This also facilitates tracing data provenance. In this case, we see that 74%, or 316 out of 422 total files, have valid persistent identifiers. Following that, the ratio of files, biosamples, and subjects which are associated with different ontology terms is displayed. These metrics tell us that 98% of files, or 418 out of 422 total files, are associated with a data type, while only 72% of files are associated with an anatomy term. The appyter also checks for these associations with biosamples and subjects. If there are defined collections or projects in the data package, the appyter also looks at the number of associations between collections, projects, and other entities. For example, we can see that 74% of the files belong to a defined collection, but only 12% of the subjects belong to a collection. Finally, we can see any ontology terms that are not associated with an entity. In this example, there are 22 assay terms that are unassociated with a file, which alerts the submitting DCC that these assay terms should be removed from the data package. An optional landing page check can also be performed, during which all persistent IDs will be checked for resolvability. Finally, the appyter provides a tabular report containing the name of each metric evaluated above, as well as the related FAIR principle and corresponding value for the input data package. This provides a unified overview of the assessment results. Additionally, a FAIRshake insignia is also generated for the input data package, which serves as a visual representation of the fairness of the data package. Hovering over each square in the insignia displays a summary of the evaluated metric. The blue squares indicate complete compliance with FAIR. The red squares mark metrics where the fairness of the data package could be significantly improved, while purple squares indicate areas that could use moderate improvement. The entire appyter, including all outputs, can be shared via a unique URL. The appyter can also be run locally using a Docker container, or be downloaded as a standalone Jupyter notebook to be shared, re-executed, or modified.